Are you able to see my slides and hear my voice?
Yes.
Great. Okay. Get my pointer here. Okay, so it's my pleasure to have the opportunity to discuss this topic with you. The output from a working group that was put together under the auspices of IQ around physiologically based pharmacokinetic modeling and drug discovery and development, and provide a pharmaceutical industry perspective on the topic. Before I really dive in, I would just like to acknowledge that I'm representing a group of folks here. It was a really great group of folks that we were working with. I really enjoyed it. We were led by Hannah Jones from Pfizer. And I think it's probably good to acknowledge that this particular effort had both an authorship group, which is listed here, but also had a reviewers group that was assembled to review the manuscript that was subsequently published and provide comments back on all that, and to give a special thanks to Scott Obach for roping us all together. So the webinar outline is essentially going to follow the same structure as our published paper, which there's some cutouts of it here for your reference. And so first we'll talk about the perspective of PBPK modeling. We'll then move on to a brief sort of chat about PBPK model structure and the parameters that we're using, PBPK modeling strategies and approaches within the industry, and we'll hit upon three specific applications, diverse applications of PBPK modeling within the industry, and we'll dive into those in some detail. We'll discuss challenges and future opportunities for the application of PBPK models, including interactions with regulatory agencies and regulatory acceptance of this particular type of modeling, and some internal best practices in education that would be beneficial to furthering this particular area of science. So the perspective on PBPK modeling. So your PBPK modeling has existed for quite some time, I believe since the early 70s. And it's very interesting. If there was a PubMed search that was done in support of this in October last year, retrieved approximately 1,300 or so references that were published in the mid-70s. However, about 55% of them published within the last 10 years, so there's really been a groundswell of interest in this particular type of modeling. One of the things that's believed to contribute to that is the development of different in silico models and the ability to integrate preclinical data into these models, making it a very attractive, in particular, a type of science that I'm passionate about, ADME science, being able to integrate that into the models to drive quantitative predictions and in vitro and in vivo correlations. We found diverse applications of PBPK models, both in the preclinical space and the clinical space, and this sort of groundswell of activity has reached the level of the regulators, and indeed the U.S. Food and Drug Administration and the European Medicines Agency and the Ministry of Health, Labor, and Welfare of Japan have all come out in support of use of PBPK modeling in various degrees and have guidances on the topic. And there have been PBPK workshops that have been organized by both the FDA and the MHRA on the topic, pulling people together and trying to establish some best practices and discuss issues that would be sort of common to the use and application of these type of models. So, and indeed, the word is spreading. Because of this, it was felt within the IQ, particularly within the drug metabolism leadership group and the clinical pharmacology leadership group, that it would be good to get some folks together and get a pulse on the best practice of the use of these PBPK models from within the industry. And that was brought forward, and it was under the auspices of the IQ, the International Consortium for Innovation and Quality in Pharmaceutical Development, and it's the output of this working group that I'll be discussing with you folks here today. So jumping right in, PBPK model structures in a very sort of general sense can be shown here, whereby you've got in a particular species, particular organism, you'll have a structure that's very similar to this, where there'll be an arterial blood compartment, and you'll have each one of your different tissues represented by different compartments here. And the compartments are fed by the blood flow using the actual physiology that's known in the particular organism. The size of these tissues and the composition of these tissues are based on known physiological information, and you can incorporate drug-specific properties such as liver intrinsic clearance or kidney intrinsic clearance to describe elimination of the drug from the system. And as the blood goes through the organs and enters the venous circulation, that too is being modeled, and it's returned around. It forms a sort of closed loop. And here in the gut, there can be very sophisticated, and there are very sophisticated absorption models that are being used to describe the very diverse and different processes that are occurring during the absorption of drugs, and those can be incorporated to drive oral predictions. These compartments and the blood flows, again, are defined by the actual blood flow rates and the tissue compositions in the particular organisms. A lot of these things that we're calling system parameters, so that would be parameters that are specific to the species or organism that you're actually studying, be it a rat or a human. A lot of these are being sort of collated and fed into the models that's been collected in the literature. Things like glomerular filtration rate, milligrams of microsomal protein in the liver, enzyme and transporter abundance, all can be incorporated into these models. One very important thing is that a key aspect to these PBPK models is each one of these tissue compartments will have an explicit partition coefficient that relates the blood concentration to the tissue concentration, and there are in silico models that have been built which leverage known tissue composition in terms of lipid content and various other factors, and that's also being used to drive the predictions here. So it's really about integrating all this information together. And one of the things that, speaking as a pharmaceutical scientist, that I can say that I find really attractive about this is that it enables us to collaborate across different parts of an R&D organization. It's quite common for us here at Merck to work with our formulation colleagues to build these type of models, and it gets us all speaking the same language, and it's very attractive, in my opinion, from that particular aspect. PBPK modeling strategies and approaches within the industry are quite diverse and start at the very earliest stages of discovery and can proceed and grow all along through late development. So in the early discovery, some of the things that we're doing with these would be early risk assessments in terms of things like, do we think we have a dose that we're going to be able to develop? Are there any potential drug interaction liabilities? Are there any potential pharmacokinetic liabilities? These sorts of things with appropriate data and integration in the models can be understood to some level at this very earliest stage. Using them to support toxicokinetic dose projections to help out our safety assessment and toxicology colleagues. And early formulation assessments are also being conducted at this stage to help us understand what type of formulation strategy we're going to need to achieve our goals in the clinic. And indeed, we're going to be seeing one of the examples that we have is in this very similar type of area. In early clinical development, the nice thing is you start building these models in discovery. Once you move into early development, the models can be challenged and can be refined through a learn and confirm data integration process. It's quite common in modeling. Things like first-in-human projections, helping to design single multiple dose ascending studies. Efficacy, PBPK/PD modeling can be done. Drug interactions, those sorts of things can be done at that early development stage, and the model can continue to grow. And when you get to the late development stage, there's types of things that can be done around design of those larger trials. understanding the PK and PD in special populations such as organ impairment, such as pediatrics. We'll be seeing one of those today. And actually, in some cases, has been influencing the actual drug labels that come out of the development study. So you can see it actually has a wide variety of uses, and the models can learn and grow with the compound, providing a nice repository of the information. It's also another very attractive feature of this particular type of modeling. So when it comes to the building and integration of data in these type of models, there's been some terminology that's developed over the past several years, I think, and we'll attempt to walk you guys through some of this. One sort of terminology that's come up are this idea of bottom-up or top-down models, and actually a third one that's called middle-out. So we'll walk through what we believe these different terms mean. In the case of a bottoms-up model, you really start with the compound data, be it from in vitro or in silico information or other preclinical type of information. And then you'll have your system physiology parameters, the things that I had described on a few slides earlier. And those are integrated together into the PBPK model. And it's a good idea to use that PBPK model and challenge it with some observed data. Say you've built the model in rat or dog. At this particular stage, you haven't went into humans. Challenge the model to see if the assumptions are holding up, and if need be, feedback or refine that as you would need. Here we've listed this as model verification. At Merck, we're advocating the use of the term qualification for this step, but I think we're talking about similar things there. Once you've got that process completed, you can then be comfortable using it for predictive applications. Of course, once that data comes in from the predictive applications, you would always want to be conscious of feeding that back in and trying to refine the model as needed. In the case of a top-down model, there's what may seem on the surface as a slight difference, but it actually is an important one, where you start with the same sort of ingredients over here. You've got your in silico or preclinical data, you've got your systems data, but there's also some observed pharmacokinetics that's integrated in at this particular step where parameters are fit to observe PK, and what we're talking about here is not really wholesale fitting hundreds of parameters. We're talking about having a hypothesis around a very small number of thoughtful parameters that can be fit to the data to actually generate the actual PBPK model, which is then used for predictive applications. And of course, the same goes here. Once those data come in, you're going to want to feed that through and kind of continuously qualify the model for the purpose. This kind of process here, where you would check and you would perhaps maybe refit or fit different parameters if there's a hypothesis that something else is off. That's, in my opinion, what's referred to as a middle-out approach, and I think that's where some of our best models will come from, is through this type of approach. Model inputs for these type of models should be from high quality in vitro and in vivo studies. It's the idea that if you start with garbage, you're probably going to get garbage out. So you want to have good quality in vitro and in vivo data that's feeding these things. As it might sort of be obvious that the knowledge of the compound of interest can grow, and this model can serve as a repository here. And verification or qualifications of the model assumptions preclinically before predicting in human is a good practice that should be followed when possible. And we can actually use sort of other pharmacometrics tools like parameter sensitivity analysis to identify influential model parameters. And those influential model parameters, those sensitive ones, if you will, could be studied with lower throughput or definitive assays to provide the best possible parameterization of whatever you're looking at that's possible. Things like intrinsic clearance or volumes or something like that. So I'm going to jump into discussing the first of the three applications that we have here, and I would just take a moment to pause to say that in the actual manuscript that was published and in the supplementary material, there's a really big table that shows a wide diversity of applications within the pharmaceutical industry. I think when we got together, we realized that the type of the number of things that are sent on to the regulatory agencies is really small relative to the amount of modeling that's being done and being used for internal decision making. We wanted to try and express that somehow. So I would encourage any of you to go and take a look at that supplementary material. There's a lot of information there, and it shows a huge diversity of applications. And we also tried to give some sort of qualitative assessment of how difficult we thought the different applications were in terms of being able to pull them off. So this one is a really nice example. This was a PBPK application in terms of developing a modified release formulation. This particular compound was a weakly basic lipophilic amine that had low aqueous solubility. The solubility was better in biorelevant media, but still wasn't great. The compound was brought forward and studied in a phase 1 healthy volunteer study using an immediate release formulation. And the team had noticed through the course of the clinical studies that there was a particular AE that seemed to correlate or trend with the Cmax, the maximal plasma concentration that was attained after dosing. And they had also observed that when they administered food, that the compound had better tolerability. And we'll see some of the data. Actually, with the food, they achieved a lower Cmax relative to fed conditions and a longer Tmax. So that led to a hypothesis that if you were able to slow down the drug release using a modified release formulation, that'll reduce the rate of absorption relative to the immediate release, producing a lower Cmax and improved tolerability. One of the important things is through whatever PK/PD information was available for the compound, the team believed that there was a certain trough plasma concentration that was required for the patients to have efficacy. So producing this lower Cmax and improved tolerability couldn't come at the expense of the efficacy. So the formulation, you wanted to achieve a similar C trough. And the other thing with this type of work is that to get good absorption all along the dose interval for the modified release, you really need to have good absorption from the distal gastrointestinal tract for this to really work out the way it's intended. So here's some of the data. In panel A is the actual healthy volunteer data. You'll see up here plasma concentration and time. The black circles here are the observed data from the fasted immediate release formulation, and the red are from the healthy fed formulation. And the PBPK model predictions are shown in the black solid line and the red dotted line respectively. And what you can see is that the model did a really good job describing the general shape of the curve and area and the salient features, the Tmax and Cmax, for example, in the fasted condition. The simulations of the fed state didn't quite capture the entire profile, but there was a couple important things that were noted here. One is that the simulations from the fed condition did predict that a lower Cmax would be achieved in that particular state, and also that the Cmax was lower and that the Tmax was extended somewhat. So the team took this model and through some preclinical work, likely involving in vitro dissolution and things like this, they thought that they could achieve release rates for this molecule that were on the order of a 5-hour and a 10-hour release rate based on the properties of the molecule and what they were able to achieve. They used the PBPK model to simulate what would that look like in a human being. And these are the simulations shown here in graph B. And indeed, the 10-hour and 5-hour release rates seem to match with the objectives that they had in terms of lowering that Cmax, delaying the Tmax, and actually having good exposure out here at your terminal point needed for efficacy. So based on these in vivo model results, the team actually went to using some targeted and model-informed resource-sparing non-human primate studies conducted to determine if the in vivo absorption rate of these prototype formulations would actually bear out in an actual in vivo study, and to compare those against the model predictions leveraging PBPK. And here are the results from that investigation. Here we've got a very similar setup. We have plasma concentration versus time profile. This is from the non-human primate study. So the team built a PBPK model in non-human primate and ran these studies, simulated out what they should look like, and the simulations worked out really well. You can see that the 5-hour release rate and 12-hour release rate respectively had the 12-hour release rate had a lower Cmax, blunted profile here, and essentially the PBPK model was able to recapitulate that to a really nice degree, which sets them up to then go on and do a very targeted human clinical formulation study where they tested the different release rates. Here there was a 5-hour and a 10-hour release rate, and what you can see is the model simulations and the actual observed profiles that were achieved, and I think it's fair to say that the team achieved their goal in terms of being able to get that better tolerability, lowering the Cmax, and achieving their efficacy out here at trough. This is a nice example because it shows how when a team has a hypothesis, PBPK modeling can feed into that, and actually with a very limited in vivo animal studies, a limited and targeted investment, you can actually bring forward hypotheses and get efficient strategies for testing these things in actual clinical development. So our second application I'm going to be talking about here is a really interesting one. I really like this one. I like all of them. I remember seeing this a long time ago and thinking this was really neat. This was the prediction of a DDI potential for a candidate drug that has nonlinear disposition. And really, in my opinion, the team were trying to hit a moving target from a moving platform here, and it worked remarkably well. So let's walk through it. This particular compound was a lipophilic amine with a pKa of around 8.2. They had some hepatic uptake studies which suggested that the unbound intracellular levels were about 3.4-fold higher than unbound plasma concentrations. All that information was integrated into the PBPK model. They had some in vitro reaction phenotyping studies that suggested that CYP1A2, cytochrome P450-1A2, was an important enzyme in the metabolism of the compound, and that was derived from studies including recombinant CYP studies in combination with an ICEF approach, studies using the inhibitor ABT, and a selective competitive inhibitor, fluvoxamine, all sort of led to the conclusion that CYP1A2 was important there. And the team actually characterized in vitro the kinetics from the recombinant system and had a KM around, unbound KM around 1.2, observed in vitro. An additional complicating factor is this molecule was also a time-dependent inhibitor of CYP1A2, and the kinetics of inactivation were characterized and they're listed here. The typical things that we look at, the Ki and Kinact, are listed. So what actually is going on here is this molecule, through this CYP1A2 pathway, actually has some nonlinearities in which the fraction of the dose being metabolized by CYP1A2 is dynamically changing over the dosing interval. And this is some of the data that we can use to illustrate some of that. So here in panel A is a plot of the liver intrinsic clearance through the CYP1A pathway versus time after dose. And you can see that after a 50 milligram dose, there's a rapid decrease in the intrinsic clearance as it recovers as the drug is kind of washed out. At a higher dose, 400 mg dose, there's a much more extended and prolonged decrease in the intrinsic clearance. It takes a little bit longer to come back. This is a combination both of saturation of the CYP1A2 enzyme and the concomitant time-dependent inhibition that was observed. It's worthwhile mentioning that the bottom-up model that was used to predict into the early clinical studies, in general, fell within twofold of the observed exposure, which is pretty remarkable from my opinion based on all the moving parts here. But the team, there was obviously some misprediction, so they middled out the model, if you will, by fitting the Km and Vmax to the clinical data using a Bayesian approach, and that yields the model that we're seeing here in panel C and D. And what's being shown here now is the plasma concentration time data from the human studies, plasma concentration and time. Panel C being the 50 milligram dose, panel D being a 400 milligram dose. The green symbols here represent the green line here represents the observed data. The red line represents the PBPK model. And you can see it's recapitulating the actual shape of the curve very well. And this model predicts that the fraction of the elimination going through the CYP1A2 will be nonlinear at therapeutically relevant doses. This is important because the team was really concerned about CYP1A2 drug interactions. It was a problem for this molecule in the therapeutic area that it was intended to go into. And the predicted drug interaction through 1A2 inhibition at clinically relevant doses was expected to be less than what was observed at a low dose. So the initial study that was done from the team was a low dose study where they had tested the model in a clinical drug interaction study using fluvoxamine. And what they saw at that low dose was a pretty substantial drug interaction here. The observed AUC GMR being higher than 20. The PBPK model actually suggested that at therapeutically relevant doses that this DDI would be mitigated somewhat. That because of the saturation of the enzyme, because of the concomitant time-dependent inhibition, that the actual clearance through the 1A2 pathway is going to be mitigated somewhat. And the modelers were able to convince the team to go and conduct a study at the therapeutically relevant dose to understand the true therapeutic risk of this drug interaction. And you can see the actual results here from that low dose, the high dose again being more the therapeutically relevant one. The predicted GMR at both dosing situations remarkably close to what's observed, both in terms of AUC geometric mean ratio and Cmax geometric mean ratio. And this PBPK, and so you can see the observed data here. We can walk through some of this. So panel A is the simulated and observed data following a 75 milligram BID dose for the compound. Panel B is the simulated and observed concentration time profiles for the low dose, the 15 milligram dose, single dose of the compound with or without fluvoxamine. The green line is without fluvoxamine, black line is with fluvoxamine. And a very similar plot at the higher dose there, the 100 milligram dose. And this was the actual multiple dose simulation that was used, and that could be compared to these data. So the PBPK model had a significant impact on this development, that enabled the hypothesis to be generated that I frankly don't think would have been terribly intuitive, nor I think you would have had challenges doing this from a static model perspective, that the observed DDI that was originally observed at the low dose probably isn't reflective of the true therapeutic situation, and that could have resulted in an inappropriate termination of the compound in development. This DDI that was observed at the more therapeutically relevant dose was considered manageable by the team. So that's a real nice application there. The third application that I'd like to talk to you about was something that we had done here at Merck, and I'd like to acknowledge Yan Zhu and Georgie Hartman, who helped helped me and actually did most of the work here. And this was around translation of adult pharmacokinetics to different pediatric populations using the drug Singulair. So Singulair is eliminated mostly by P450 biotransformation in human. There was a study that was published using this recombinant ISEF approach that we had used and integrated into the model that suggested that the fractional intrinsic clearance through the different enzymes could be parameterized in the following manner: about 72% through 2C8, 12% through 2C9, and 16% through 3A4. And the reason why this is important and really critical to achieve the goal of pediatric translation is because when we can parameterize the model in terms of the individual enzymes, and well, I would just say individual processes, enzymes, transporters, or anything else really, that it allows you to model the specific mechanistic ontogeny or development of those pathways as they go on. If you don't parameterize the model that way, you really can't exploit that previous knowledge on the ontogeny of these things. There was a trace amount of some glucuronidation that's observed for Singulair, but it's very insignificant relative to the P450. And there was a measured minor contribution of renal excretion that was also incorporated into the PBPK model. And what data did we have? We had adult intravenous and oral PK data available that was used to both build and qualify the model from fed and fasted states and different oral formulations. The metabolic clearance was estimated by subtracting the renal clearance from the total body clearance, and once we had that estimate of the metabolic clearance, the intrinsic clearance was apportioned using these percentages listed up here using the recombinant CYP data, and that is how we actually parameterize the model. I should say, even though I'm not showing it to you, if we just put the recombinant CYP data in a bottom-up approach, it actually came pretty close to the observed data, well within twofold, but we wanted to get this as close as possible. So we basically, in essence, you could say we fit the clearance of this particular model, then apportioned it out this way. And I'll say that that was the only fitting that was done for this entire process, and the rest of it was complete simulation. Basic ADME and formulation and physical chemical incorporated into reduced PBPK model structure. And again, we built and qualified using adult pharmacokinetic data before simulating the pharmacokinetics in children, consistent with current thoughts from the regulators and various academicians on the use of these models for this particular purpose. I think that's a really good way of doing it. So here's actually some of the data and the output. In panel A is again, this was the model building step. Okay, so we had fit, if you will, the clearance and apportioned it as I described to you. We could see the observed data and the model predicted data here. This was following a 7 milligram IV dose. We then used that model to then go on and simulate these different conditions. We have a 50 milligram oral solution that was given to adults, as well as a 10 milligram film-coated tablet, both in the fed and fasted states in panels C and D. And when we looked at the agreement between the observed data and the model predicted data, we thought we had a reasonably well described the actual profiles here with the PBPK model. All key metrics are well within twofold of the observed. In fact, they're much closer, both in fed and fasted condition, even with the solution and the film-coated tablet. So we, at the time, we considered this qualified and then went on to simulate the pharmacokinetics in the children. There's a lot of other data we could have used to qualify this approach. This is, in the interest of time, this is where we had stopped. So, and we'll walk through this. And so here's the actual simulation of the observed pharmacokinetic data in different pediatric dosing groups. So let's walk through these. Panel A is a 4 milligram oral dose as oral granule formulation in 1 to 3 month-year-old children. Panel B would be 3 to 6 month-year-old children. And panel C is 6 months to 2 years old children, all with that oral granule formulation. Panel D is a 4 milligram film-coated tablet to 2 to 5-year-olds, and that same formulation in 6 to 8-year-olds. And the last panel is a 10 milligram oral dose film-coated tablet, again, to 9 to 13-year-olds. So using the specific formulation, the different characteristics of these formulations in terms of particle size and dissolution characteristics, and the model that was built and qualified using the adult data, did a very nice job simulating the pharmacokinetics of the drug in all these different populations of pediatric individuals. There was some misspecification here on the absorption in some cases, and that's because, well, at least maybe partly because some of the pediatric GI physiology is not really well understood. I think it should be obvious why that it is. We're using some adult data, or in some cases data that's been translated down in some manner to inform the system parameters for the pediatrics there. The model predicts that, in general, that the fraction of the dose that's given generally decreases with age, and also suggested that there's no major changes in the fractional clearance by the CYPs with age, even down to these young levels, that would really sort of say that there's no unique pharmacogenetic or DDI concerns. It's also something that's quite unique to PBPK models. I don't think that the typical kind of allometrically scaled POP-PK model would be able to inform on either one of those two aspects. So challenges and future opportunities. There's clearly many challenges and many future opportunities. Most of us believe that leveraging in vitro data and being able to model transporter-mediated uptake and elimination remains a challenge, remains an area of development. There's been some recent movement on this in recent years. It's just a matter of time before we as a scientific discipline figure out how to do that. And that really kind of speaks to being able to model free intracellular levels there. We're still working on non-CYP enzymes. We know a lot about cytochrome P450s, their induction profiles and things like this. Less is known about those non-CYPs, and people are actively working on that. A very important one is the impact of disease states. You could build one of these PBPK models and then use it to help you understand risk in, say, renal or hepatic impairment, for example. And that remains a challenge. Some of the underlying pathophysiological changes that are ongoing in those different disease states, that's still an area of development. There's actually groups out there who are studying such things right now. Future opportunities would be things like there's this Orbido Consortium that's focusing on absorption modeling. That's a nice effort that's ongoing to help understand the biopharmaceutic aspects of these PBPK models. Another area that's really been growing recently is the PBPK modeling of monoclonal antibodies. I could see PBPK models being used in the discovery and development areas for those monoclonal antibodies moving forward. I had mentioned that the regulatory agencies are very busy in terms of guidances and trying to really figure out how to best leverage these PBPK models, and increased usage of these will be a future opportunity that we in industry can partner up with our regulatory colleagues and really try to settle on what is the best use of these models and the specifics of collaboration there. And there's rapidly improving software tools and an increasing level of expertise among preclinical scientists, clinical scientists, modeling and simulation scientists, all allowing us to collaborate together, and I think it's only going to get better from here. And I hope that I've convinced you that PBPK model does have a place in the science that we do, that it facilitates a collaborative working between different disciplines, different scientists within pharmaceutical companies, with regulators, with academicians, to deliver impactful decisions and optimize the way we do drug discovery and development moving forward. last but not least, I'll just kind of show this up here while we take some questions. I want to acknowledge again Hannah Jones, who helped us lead this group. I have her email on here. You could follow up with myself or her offline with any questions that you have. I'm sure that would be fine. And I appreciate your time and interest in this, and would be happy to field questions at this point.
Thank you very much, Chris. Great presentation. We have a couple of questions here, and I do invite all of our audience to submit questions in by typing them into the question module at the right-hand side of your screen. I'll go ahead and start the questions now. Is choosing the model based on the product or disease area, or what exactly drives to choose one of the models? I'm not sure that.
So I think if the question's around, and by the way, I would invite any of my fellow authors on here to jump in at any time. I think I'm not terribly understanding the question real well, but if the question's around what type of model structure to pick, I think it really depends on the question that's being asked. So in the case of the singular example, we did use a reduced model there where a lot of the organs are sort of considered one compartment, and that was because there wasn't really any sort of specific, we weren't really asking questions around, what would be the effect, PK/PD effect in the lung, or what would be the liver uptake or something like that. We didn't really have those questions for that particular application. So I think you really need to understand what the questions are and set the model structure up accordingly. If you really need to know the concentration time profile or extend that to the pharmacokinetic, pharmacodynamic profile in those organs, then those things need to be incorporated into the model.
Okay, great. Thank you. Another question is for translation of adult PK to pediatrics. Did you take into account the dynamic changes in CYP maturation in pediatric population? How was it incorporated in the model?
Yeah, that's a real good question. So how it's being, there's abundance, different groups are generating data, and I actually hope, this is a plea to the community to please give us more of this. I think we need more of it, frankly, where they've been measuring the abundance of these different enzymes and transporters in pediatric different age groups when we're able to get those samples. And what you can do there is you can construct a model which basically would, in essence, have either the abundance or the activity modeled as a function of the adult. So at the adult level, you'll have 100% of the activity, and accordingly, as you go down in age range, there will be a decrease in the activity or the abundance of that enzyme as you go down to different age ranges. So, for example, I don't have the specific numbers here with me. I'm actually going to be talking about this at Land O'Lakes next week, but let's say that a 6-month-old has 50%, I'm just throwing out some numbers here, 50% of the activity relative to an adult. That information on the ontogeny will be incorporated into the model. The intrinsic clearance through that enzyme will be reduced accordingly.
Thank you. Another question is there effort to merge PBPK and PK? Thus, the nonlinear mixed effect of PK/PD can be studied in PBPK as well.
Yeah, it's an interesting question. I know there's a lot of discussion about this. I think my opinion is we're actually kind of studying similar things, but in different ways. We're actually speaking a lot of the same things, and many times, what I've seen in meetings is we're talking past each other. I'm not so sure from a modeling perspective how to merge those two together. in general, PK models would be more your top-down approach, where you would be introducing different covariates into the models and trying to study how those covariates relate to the metrics of interest, if it's PK or PD. On the flip side, the PBPK is taking a slightly different, yet I would argue analogous approach, whereby we're trying to assign mechanistic reasons for why would the pharmacokinetics of a compound be different in a healthy population versus an hepatic impairment population. Mechanistically, why would that be the case? I think that's where PBPK comes in, and I don't know if I'm doing a very good job answering that one, and would invite any of my fellow authors to chime in on that if anybody had a comment. I think they're actually somewhat analogous.
Thanks, Chris. Another question is, what does biorelevant medium exactly refer to? PBS, phosphate, I'm sorry if I mispronounced that, buffer, or any other buffer?
Yeah, that's a good question. I don't know if Neil, Maha, are we able to unmute certain individuals? I think it'd be better to have Neil discuss this one. Neil Parent, if he's on.
He's actually not on the line.
Okay, fair enough. I can speak to it then. That was actually his example. So, biorelevant media would be things. We deal with this like simulated gastric fluid, and they have something called FASIF, which is fasted simulated intestinal fluid, or FESIF, fed simulated intestinal fluid. Those things will have what's believed to be physiologically relevant concentrations of the different salts. The pH will be physiologically relevant. There'll be some bile acids that are put into those media. The idea that it's a little more physiologically relevant than studying something, say, in phosphate buffer or just pure aqueous solubility. You try to put those things into that media that actually are probably present, or not probably, are present in the body, and try to understand the solubility of the compound under a physiologic condition. And for a lot of sparingly soluble drugs, you can actually see that even the small amount of the bile acids and those things that are in that actually can improve the solubility can be higher in those relative to something, say, a typical PDS solubility, for example.
Okay,
so for anybody more about that, you look up the simulated gastric fluid or FASIF and FESIF, and you'll learn more about those. I think the recipes are somewhat standard as well, but that's not my particular area of expertise.
Thanks, Chris. A question from Lee Yu asking about the second example. Can you clarify why the DDI is more severe at lower dose than higher dose? Is this for the drug X as a victim of CYP1A2?
Yes, it is as a victim of CYP1A2. Is Jan on the line, Maha, Leah?
Let me check. No.
Okay.
So what is the last name?
Snowy. I think it's S-N-O-E-Y.
No, I don't, I'm afraid.
Okay. That's okay. I can speak to it. I understand this one pretty well. I was really sort of attracted to this particular example. It is about the victim. So the molecule here that they're referring to is the victim of a CYP1A2 mediated drug interaction. And the reason why that at the low dose, here where the drug interaction was pretty severe, the two things were going on. One, I mentioned to you that the molecule in and of itself is a time-dependent inhibitor of CYP1A2. So it's going to, there's going to be this, some people call this auto-inhibition, for example. So the higher the drug levels get, it's going to inactivate the CYP1A2 enzyme. And on top of that, in this one, there was also predicted to be some saturation, just typical Michaelis-Menten saturation of the CYP1A2 pathway over the dose interval. So at low doses, the fraction of the clearance of this molecule going through CYP1A2, which is the affected pathway here, is greater than it is at the higher dose. At the higher dose, there's more inactivation of the CYP1A2, removing it from the system, and there's also more saturation of the CYP1A2. So the fraction of the elimination going through that pathway is less, and that is the reason why at a higher dose, again, a therapeutically relevant dose, the DDI is significantly mitigated at that point. It's because the fractions change.
Thank you. Diane Ramsden has another question. In order to have accurate DDI predictions of CYP3A4 substrates, is it necessary to have a good estimate of FG, the fraction of escaping gut metabolism? It has been published that simulations of FG utilizing readily available PBPK software, in vitro liver, metabolic, and permeability data can be used for estimations of FG instead of performing in vitro experiments. 1, can you comment on the frequency of such predictive techniques in your labs? And 2, on the accuracy of predictions obtained using these simulated values?
Yeah, I mean, I can comment on my particular experience. I can't comment on the industry, although I will say that there is an IQ effort that is, I believe, going to be producing a manuscript in the not too distant future around victim drug interaction prediction, and I believe that that's what we're referring to here. We're talking about victim drug interaction predictions for 3A4 substrates. Yes, it is my opinion that you do need to understand the gut metabolism there. The way I think that things work, that is a key component, and I think there's some clinical and modeling information out there that supports that. In terms of the frequency, you know, I can only speak from my own personal experience. It's not too frequent yet. I mean, we've started doing some of that where we would use this preclinical data to parameterize the model, make some risk assessment for the victim drug interaction potential. We've got a handful of these, and they seem to work okay. I'd like to get a little more experience with it before sort of really kind of casting any kind of judgment one way or the other. And I would just point out that even a lot of the parameters that you would be needing to generate to drive a PBPK model prediction on the victim side can also be incorporated into static models, so to give you some kind of risk assessment. Though you'll find that when you dig into it, the parameters are very similar there. So yeah, to sum up, you know, it's not a standard thing, I could say, but I hope to see the usage of such tools when it's relevant, being used more in the future, and we have had some success with that.
Okay, thank you. Fang Wu has a question. You mentioned that there could be absorption prediction error in using PBPK in pediatrics. Could you please give further explanation on that?
Yeah, it's a really good question, and in one of my internal slide sets I actually had. It's been a long time since I've looked at that, but I think some of the physiological parameters, and I can't speak to specifics here, but I can give you maybe some generalities in terms of what maybe we're looking at. You know, one would be things like the transit times through the different segments of the gastrointestinal tract. You know, may not be completely well understood in the different pediatric populations. Potentially things like the abundance of the enzymes, the abundance of transporters along the length of the gastrointestinal tract may not be totally well understood at this point. pHs and things like that. It's just maybe it's not so obvious, but it's not really easy to get that type of information from a pediatric population. We're really left trying to piece things together as best we can.
Okay,
so it would be those system parameters.
Okay. Linkley Lee has a question. What are the parameters in the SIM-CYP model to reflect the saturation and time-dependent inhibition of, oh, wait a minute, I'm sorry, my question just disappeared. Let me actually ask a different question so I can find that one. I apologize, Linkley. For transporter-mediated DDIs, for example, OAT1, P1B, I apologize. What's your view on scaling factor? Is it possible to have a universal scaling factor applied to different substrate drugs?
Yeah, that's right now that's the holy grail. Well, I mean, I've seen some work coming from Tristan Mauer's group that suggests that it is possible to have a single scaling factor. I think there's a lot of unknowns still there, particularly around the liver concentrations. It's one thing to drive a plasma prediction when you've got these uptake transporters. It's a whole nother to really understand the liver levels, particularly when you've got a therapeutic that requires those liver levels for efficacy, like an HCV drug, for example. I don't know if it will ultimately be possible to do that. I think we need to study it more. I mean, there is some suggestion that it is possible. The situation, in my opinion, is not that dissimilar, really, to the ISEF type approach that we're using for recombinant CYPs. There's some, data that continues to come out on the utility of that. So I'm not real sure. I would like to. I don't know if it's going to be one, the one scaler that fits all, or if there'll be different scalars. But I sure look forward to the day where we're able to take in vitro data on transporters and be able to integrate it in a quantitative manner into these models. I don't discipline. We're going to make a lot of huge strides forward, really, in terms of predicted ADME and predictive disposition until we're able to do that. That's just my opinion.
Okay, we'll go back to Linkley's question. She has an additional one, so I'll read them both. What are the parameters in the SIM-CYP model to reflect, I apologize, there's the same question. What are the parameters in the SIM-CYP model to reflect the saturation and time-dependent inhibition of CYP1A2 in your second example?
Yeah, it's a good question. So what they, I wasn't showing at all. What the team did here was they generated enzyme kinetic data in recombinant CYP1A2. So not only did they have a KM here, but there was also a Vmax, obviously. So if you put the KM and the Vmax in, and that's how you're parameterizing the intrinsic clearance or the metabolic clearance, as it would be through the pathway. If your unbound liver levels are, start to approximate 10 times above that, you're clearly going to be in a saturation range. So it's the KM and Vmax that can drive that part. In terms of the time-dependent inhibition, which is also feeding back on the pool of enzyme, that is characterized by these time-dependent inhibition parameters called Ki and Kinact, and those can be generated in vitro and incorporated into the model. So it's really a combination of kind of a traditional enzyme kinetics and pretty well established kinetics for time-dependent inactivation that are both put together in this particular case to model that nonlinearity. If you just parameterize the model in terms of an intrinsic clearance, you can't drive any saturation with it for sure. I mean, you really need something, some hyperbolic function there to do that, and that's how it's that's how it was achieved in this case.
Thanks, Chris. King Verdi has a question. To generate a PBPK model for pediatric formulation, is it critical to take into account the significant difference in diets for the pediatric population?
Yeah, that's a real good question. You know, that would like speak to food effects and stuff. And you know, I'll be real honest with you. I don't have a whole lot of experience there. I have 2 young children myself, and their diets are pretty different from mine. I can tell you that, so I can appreciate the comment. But I don't have a lot of experience with food effects there, and what parameters or considerations need to be accounted for in that particular application. I can't really speak to that.
Okay. Matt Covey has a question. I am an analytical scientist and just want to understand when it comes to working with pediatric formulations, what is the good time to evaluate or extrapolate adult models towards pediatric modeling assumption? And also, is it good to actually perform studies using FASIF and FESIF medias or extrapolate the data?
I'll answer the second one first. And again, these are just my feelings, but I think you're doing yourself a disservice if you don't understand the solubility of your molecules in those biorelevant media. Definitely, that's a value-added endeavor, in my opinion. And in my opinion, also on the time to do these things, I actually, yeah, I have a pretty strong opinion about that. And I think that, in fact, and we've started to do this, it was part of our retrospective study here at Merck, that the type of model that you would set up to understand victim potential could also be used to simulate your pediatric would look like. There's really that, you know, there's really nothing stopping you from the very earliest clinical development, in early clinical development, in terms of once you've got some human data to help qualify the model, there'd be nothing stopping you from running those simulations, as long as the model was parameterized in a thoughtful way where the individual processes involved in the elimination of the drug can be modeled. It is my opinion that that should be done. I mean, if you're waiting to phase 3 to do that, it's my opinion that's too late. You could be doing it a lot earlier than that and thinking ahead.
Thank you. What will the FDA preference in between these 2 stages? What will the FDA preference in between these 2 stages? I'm not sure I understand the question.
Yeah, and I can't speak to FDA preferences. I wouldn't even attempt that.
Okay, thank you. So I think we've got quite a few more, so we'll continue. Is the intrinsic clearance obtained from your SimCip model under different dose regimen condition in the second example has reflect both saturation and time-dependent inhibition, or only the first one, saturation?
No, both effects are being modeled simultaneously. So you can see here at that higher dose, there is a more pronounced decrease in the liver intrinsic clearance. It's also you could notice that it's sustained for a much longer period of time. That's not only due to the saturation, but also the fact that the molecule in and of itself is inactivating the enzyme. So both effects are being modeled here simultaneously.
Thank you. What was the rationale supporting the decision to do parameter estimation for CYP1A2 KM and Vmax in the example of nonlinear disposition, as opposed to doing the same for Ki and Kinect? I ask because I would think that the inactivation plays a significant role in explaining the decreased CYP1A2 component of clearance at higher doses.
Yeah, that's a really good comment, and I sure wish Yan was here to to address that. I can't speak to why they did that particular one. I had a similar thought myself. I think you could argue that you know, you could do one or the other. I think it would be a mistake to fit both at the same time, and that's one of the things that we did mention in the manuscript, that when you start to overfit these models, you can oftentimes get into a situation where there are identifiability issues, and I think that compromises the predictive performance of these, and can even, in my opinion, compromise the confidence in the model. So for one reason or another, this team had a hypothesis that this would be where they would want to fit the model to middle it out, and they didn't fit the KI and KNAC data. It could have been a case where, and I'm purely speculating here, that maybe there were some issues with the KM-VMAX study, or maybe their data were more solid for the time-dependent inactivation parameters. I can't really speak to that, but I think that strategically it would be a situation where you would want to look at those data sets and pick one or the other to model here. And maybe, for all I know, there were sensitivity analyses that were done that suggested that was where the biggest nonlinearity was. I mean, it's hard to say, but those are my suspicions.
Thank you. Do you recommend doing monkey PK study before human BA study for modified release formulation?
I mean, I don't think there's anything sort of magical about monkeys or any other species. We have used we've had very similar kind of activities here at Merck, and we use dogs for those. There could be situations where, for a given therapeutic area or a given pharmacological target, you might not be able to dose a given therapeutic to monkeys or to dogs because of some toxicity. You may be forced to use one or the other there. So I don't think there's anything sort of magic or locking you into a non-human primate study in that case. The more important thing was that that they had a hypothesis going in that those modified release formulations would achieve the PK profile that they wanted to. They used what they were able to achieve in vitro to simulate out what they would see in the monkey before they did the study. The simulation suggested that, yeah, this strategy will work. They qualified that in the monkey study and then used that as a justification for the human. What preclinical species you use to help build confidence and to qualify the approach, I think, will be up, to the individual therapeutic. But I think it would be important to do some qualification preclinically before you go testing things clinically.
Thanks, Chris. There continues to be use of allometry for human PK predictions in many circles, even for metabolized molecules. What is your philosophy regarding this, and do you primarily use PBPK for pre-FIH predictions of PK and therapeutic index or dose projections in your work for metabolized molecules, or do you also use allometry?
Yeah, so it's an interesting question, and I think there might even be a little bit of a misconception here. I mean, you can parameterize one of these PBPK models with an allometrically scaled clearance prediction. I mean, there's nothing stopping you from doing that whatsoever. My philosophy, and this is based on some internal data we have. Again, this is just me speaking here. For low clearance compounds, it seems that our more in vitro based models for low intrinsic clearance, low extraction ratio drugs seems to work a little better in terms of a quantitative prediction than allometry. It's not to say the allometry is terrible. In some cases it is terrible, but in most cases it's not terrible. It'll get you in the ballpark. But those in vitro methods seem to work a little better for those low extraction ratio drugs. And even if you are using allometry, again, it can be incorporated into the PBPK model just as easily as in vitro data. In terms of how often PBPK models are used for PK predictions in Merck, You know, I can only speak to my experience here. It's not terribly often that we're using a physiologically based model for those things. You know, that's something that we're going to start looking at a little closer. But it's certainly not commonplace to be using them at this point. We really use these PBPK models when there's a specific question that we believe this model would be uniquely poised to address. As I think one of the challenge or the future opportunities that we have around rapidly increasing level of expertise among scientists within the pharmaceutical industry, which I've personally seen myself. You know, it won't be such a huge barrier or a huge uphill battle or something like that to begin using these models even earlier for things like first-in-human prediction. I think there's a place for that, but it's in my experience not totally commonplace just yet.
Okay, thanks. Recent publication shows that singular is mainly metabolized by UGT instead of CYP2C8. How is your simulation translated to pediatrics?
Maybe there's some new information out there. I mean, there's human ADME data on that that's been around for a while, and I don't think the UGT was a major component there. So I don't think that that's true. There is some direct UGT conjugation, but it was a relatively minor pathway from the human ADME study that we have internally, and I believe has been published. I know it's been published. I'll have to do some digging to see if there's something new on there, but the UGT component was not accounted for. When I looked at the data, it wasn't a significant pathway. So that was an assumption that we made, but there was a pathway that was missing.
Thanks, Chris. What is the relationship between hepatic intrinsic clearance and the clearance observed from clinical data?
Oh yeah, I think these things, you know, something like the well-stirred model or parallel 2 models, you know, those models can show you how intrinsic clearance relates to total clearance or hepatic clearance that you would see from a clinical study. So I would encourage you to look those things up. The specifics of how the intrinsic clearance has been incorporated into the models. There's a nice publication by Karen Rowland-Yo from SimSip showing the actual structure of the PBPK model and how the intrinsic clearance is fed into that one. The well-stirred model really is a steady state relationship that only applies when there's no changes in the system. So if you've got a situation like what we're seeing here where you've got a changing intrinsic clearance over time, that well-stirred model will break down. So it's implemented slightly differently here, but you know, the unbound concepts and things like this are still being incorporated.
Thank you. How successful have you been in predicting PK in Asians, for example, Japanese, using commercially available population PBPK MNS software?
Yeah, it's a good question. I see that as an opportunity for pharmaceutical industry and you know our Asian regulatory colleagues. We've done this a little bit. We haven't had, and maybe I might be missing some literature, so I apologize if I am, but I haven't seen a real kind of detailed, systematic study with a lot of case examples. We've got a handful of them, a small handful, I would say, here within Merck, and I think our Japanese colleagues within Merck are actually doing some more evaluations on that to try and figure out when we could say you develop a PBPK model with adult data from a different population, say a Caucasian population, and then translate that to an Asian population, say Japanese, and perhaps use that information in a with all the proper qualifications and whatnot, and use that to potentially avoid some early studies in Japanese population. I mean, that was really the objective there. And I have, my personal experience is somewhat limited, but I would say is encouraging. I would leave it at that.
Thank you. Okay, we have just a couple more questions here. There continues to be, hang on just a moment. There continues to be, okay, so that one. I think we're actually all set. We're all finished. If we have any other questions, I recommend that we finish this up. We have about 7 minutes left, and we're happy to entertain additional questions until then. If we don't have any additional questions, Chris, I wanted to let you know that I think this has been a really great presentation. Certainly people can get in touch with you via your email, and Hannah's on the end, at the end of your slide deck there. If you have any additional comments before we close, welcome you to have them now.
No, I mean, they were all really, really good questions. I need to dig around to see what was published on the Lucast. But I think very insightful questions, and I really appreciate the opportunity, and just thank you again.
Wonderful. Thank you all very much for attending this webinar, and I look forward to you participating in future. Thank you.